Okay, I just wanted to remind you that you're supposed to make sure you read your lab so that you can collect the right samples for this week's lab. And again, your GSIs, consider the collections that you bring into the lab part of the lab exercise. And so please respond and take some time to find these items that we ask you to find. It'll help you to think about them and to place them in nature. Last time I started talking about plant development. And plant development is a fascinating topic. There are all kinds of aspects that most of you have never heard about before, and there are ways in which plants respond to their environment that are just totally miraculous. And what we want to do today is to continue to talk about the plant body and to introduce you to a few of the cell and tissue types which will help us to appreciate, hopefully, and to understand better the controls of plant development. Now, last time I ended by talking to you about the embryo axis. And we said that the embryo axis consisted of the cotyledons, if it's a eudicot, cotyledons, and two regions of high mitosis, or cell division activity. This regions of cell division activity is located at the tip of the root, as we mentioned last time, and the tip of the shoot. Now, we call these regions of cell division activity or high activity meristems. And we said that there was one located at the tips of all roots, which we said was the root apical meristem. And one located at the tips of all shoots, which is the shoot apical meristem. Now, following embryonic development and the establishment of the embryonic axis, what we can say is that new cell production is confined to the apical meristems. Now, the cells which make up the apical meristems are remarkable. We think that as long as they aren't pathogenized by some bacterium or some kind of plant disease, as long as they're provided food and water, that the cells which make up the apical meristem, we think, can live forever. So potentially on a 3,000-year-old redwood tree, you could have an apical meristem which was around at the time the seedling was starting 3,000 years earlier. And in this sense, the cells which comprise the apical meristem are viewed often as having properties of animal stem cells. That is, having unlimited properties to divide and, as we will see, to differentiate. So this is sometimes applied to cells of the apical meristem. The other thing we can say about the apical meristems is that apical meristems are primarily responsible for lengthening the shoot and root axis, or axes. And how does it do this? How does it lengthen the root and the shoot axes? It does that by producing cells which will elongate the apical meristems contribute to a lengthening of the plant axis. And let's explore this statement a little bit more. Let's take a plant which is relatively short as far as its axis is concerned, and we'll say that this is at time 0. Here's the axis of the plant. And so we can have a reference point on the plant. We'll put a ribbon or a mark, a reference point. You can put an ink mark on the plant, a reference point. At either end of this axis, we have the shoot apical meristem and the root apical meristem. And now let's wait for some time to pass. And what we find is that our reference spot is more or less at the same place, but that the axes have elongated. That is, it's gotten lengthened. This elongation of the axis will continue with more time until you can have a very tall plant, again with our reference point on the axis. Now, how does this lengthening go about? Again, cells of the shoot apical meristem or root apical meristem make new derivatives, they make new cells, and these new cells then undergo elongation. And in elongating, they essentially push the root deeper into the ground and elevate the shoot higher in the air. So the apical meristem is involved primarily with elongation growth. Every branch that you see on the tree, if it is to elongate, has to have an apical meristem on it. If we break off the apical meristem from this branch, as I've just done now, this branch will stop elongating. Every root on the bottom here, if it's to elongate, has to have an apical meristem. If the apical meristem is destroyed or somehow interfered with, that root stops elongating. So cells of the apical meristem are exceedingly important in establishing the axis of the plant. Now, we said last time that the new cells that are produced from the apical meristem are primary. Primary cells are produced from the apical meristem or apical meristems. And we call this primary growth. So through the activities, the cell division activities of the apical meristems, new cells are produced, and these are primary cells because, you can go back to your notes from last week, their origins can be traced to the apical meristem. This is a very important concept, this idea of primary growth and of apical meristems. Today, what we want to do is to talk to you about the primary plant body. And to talk to you about the cells and the tissues which make up the primary plant body, cells and tissues which arise from the apical meristem. So the cells and tissues arising from, let's just say, the shoot apical meristem or the root apical meristem. In order to begin our discussion, I just want to define for you a tissue, because we're going to be talking about tissues. A tissue can be defined as consisting of one or more cell types distinguished by structure and or function. So today we're going to be talking about the cell types which make up the primary plant body. And many of you are going to say, why do I want to know about the cell types that make up the primary plant body? And what I want to say at the outset is, every day, you in the audience have interactions with many, if not all, of these cell types which make up the primary plant body. They occupy many, many of the products that you use, and I want to tell you about them so that you can appreciate where they come from. The first cell type which we want to talk about is called parenchyma. This cell type is believed to be sort of the original product of early evolved apical meristems. Let's talk about the characteristics of this original cell type. One of the most important characteristics, and one that we're going to be referring to when we look at the other cell types, is whether or not this parenchyma cell, when it functions, is alive or it's dead. And in this case, a parenchyma cell is alive. That is, it has a membrane. Another characteristic is that it has thin walls. A functional characteristic of the parenchyma cells that many of you deal with, if not daily, at least weekly, is that it is a site for starch storage. And if you look at a potato essentially consists, after you get past the skin on the outside, entirely of parenchyma cells, which are filled with starch. So these parenchyma cells serve an important function as a reserve area for starch. The fourth characteristic of parenchyma cells is that they are sites for photosynthesis. A fifth characteristic, which only comes into play under some circumstances, is that they can divide to repair wounds. So, for example, if a tree is cut or a car runs into the tree and makes a gash, that hole or that injury made by the car, for example, will be filled in, will be covered by parenchyma cells which are stimulated to divide. And last, parenchyma cells, although not least, parenchyma cells participate in short distance transport. And this would be of water or nutrients. So this very important cell type is the basis for all the other cell types that we're going to be looking at today. The second cell type which we want to consider, we believe, was evolved from the parenchyma cells and is in many respects exactly identical to the parenchyma cells, except one major difference. And the second cell type is known as cholenchyma. It is very much like parenchyma, except one important difference. It has variously thickened cell walls. And it functions mostly in support. An example of cholenchyma cells that most of you are familiar with who eat celery. Here is a stalk of celery from this bunch over here. When you eat celery, you're actually eating the petiole, the part of the leaf which is not expanded. The blade or green part is something that's usually thrown away. And when you eat celery, many people will take the celery and pull off these strands on the back here. These are cholenchyma strands. These are strands where you have thick walls which give support to this petiole, which allow it to maintain a large surface area. So here cholenchyma is functioning in support of this petiole and ultimately of the entire leaf. So when you throw away these strands, you're throwing away strands of cholenchyma, and if you're an architect or a mechanical engineer, the place you would put your structural support if you were putting up a building is on the outside, the perimeter or periphery. and that's exactly where you find the parenchyma strands placed, on the periphery. The third cell type which we want to talk about is actually made up of several different cell types, and like the parenchyma cells, evolved from parenchyma. This third cell type, again, which many of you have had experience with, if not daily, at least weekly, is called sclerenchyma. And it consists of two separate cell types, one of which is very familiar to you, at least by name, and the other is not so familiar to you. The one that is very familiar to you, the cell type which is sclerenchyma, is called a fiber. And the other type, which you haven't heard of, but which is also sclerenchyma, is called a sclerid. Now, both of these function in either or both, either protection or support. We'll talk about this in a moment. And can be alive or dead when functional. Knowing the origins of the word sclerenchyma will help you to understand what these cells do and what they're made of. The word sclerenchyma comes from the Greek word skleros, which means hard. These cells are made hard by the depositing in the cell walls of a phenolic compound known as lignin. Now let's talk about the first of the two cell types which comprise the sclerenchyma. And that's the cell type that most of you are familiar with. Let's talk about a fiber. Fiber is an elongated cell with tapering end walls which overlap other fibers. And they occur in bundles. So if we were to draw a fiber for you, it would be an elongated, narrow cell with tapering end walls. We could say it's narrow. And that end walls would overlie other fibers. So here's one fiber. Here's another fiber. Now, fibers have many, many important functions in human existence. A prime function, many of you have seen this yourself, is rope. Rope consists, this is rope from a cannabis plant. Rope consists of fibers, which are extracted from the plant and then woven into the structure, which is rope. And for those of you who can come up or know from your own experience, the rope is gritty. It's coarse, actually. And that's because of the lignin, which is deposited in the cell walls. Another example of fibers that interact with us, most of us, daily, and has been exploited by humans for millennia is linen. Linen is a fiber, and it comes from the linum plant. Linum. Here we have a linum plant up here in front. This has been used, going back probably to earlier than the Egyptians, for the production of fibers for making cloth. Now, we said that fibers have sclerenchyma in the cell wall. You saw that rope up there in front. These linen fibers eventually will develop sclerenchyma in the cell walls. But until they develop sclerenchyma, they are considered to be soft fibers. And as a result, fine linen can be made from the fibers of the linum plant and not be scratchy or coarse because the fibers have not yet lignified. So lignification has been delayed in these particular fibers. Here's an example up here in front of the way fibers function in support and in giving shape to an organ. This came from a squash, something like a cucumber, but much bigger. And in this particular cucumber, what was done was to, either through natural means or through artificial means, to digest away all of the organic material which could be digested either by microbial action or by perhaps an acid, leaving behind the lignified fibers. The lignified fibers make up this structure here, which you know of as a loofah. Sometimes it's called a sponge. Of course, it's not a sponge. A loofah. And this loofah consists of fibers, essentially, which give you the shape of the fruit that they came from and can be used in washing or showering because it has a slight abrasive action due to the lignin that's deposited in the fibers of this loofah. So fibers play an extremely important role in our existence. Now what I want to do is to show you the third type of cell, a second type of cell which makes up the sclerenchyma, and to spend a few minutes telling you about its importance. And that second type of cell which makes up the sclerenchyma is called a sclerid. Least familiar to you as far as its name, but I guarantee you very familiar to you with regard to things you have eaten. Sclerid cells tend to have very thick walls. They function in protection. And they are usually small and roundish, as we will see in a moment. Now, sclerids, like the fibers, have lignin deposited in the cell walls. They are sometimes called stone cells because they are round and small and because they're hard. You have experience with sclerids in any time you eat a pear. The grittiness of the pear is due to sclerids that are part of the ovary, the fruit wall. If you eat fig Newtons, anybody here eat fig Newtons? Fig Newtons are crunchy because of the sclerids that have been deposited in the seed coat. And at one time, they found a mutant of a fig which did not make sclerids in the seed coat, and people would not eat the fig Newtons because they were used to having the crunchiness when they ate the fig Newtons. Here's an example of how the sclerids protect the plant. The inner wall of the ovary of walnuts, for example, is hard, and it is hard because it is a massive definition of sclerids, thereby protecting the embryo that's on the inside. Let's now spend a few minutes looking at a couple of these cell types. Oops. Just want to go here. Here we go. Okay. Our first cell type is of the shoot system, and here you can see the regions of the apical meristem. And if you could lower the lights, that would be great for those lights here. Thank you. Here is the shoot apical meristems located at all the tips. If you remove the apical meristem, the plant stops elongating that axis. Here's a parenchyma cell. Parenchyma cells don't seem to have much to speak for them. In these cases, you can see the thin walls. And in the next slide, you have parenchyma cells now which have starch deposited inside of them. And in a potato, the entire cell would be jam-packed with starch. Parenchyma. Here we have parenchyma now, which is, here's the cell. It's deposited in the corners of the cell and strengthens the corners, which are the weakest part of the walls. Here's another type of parenchyma, which appears toward the periphery of the plant again in sheets. And if we look at it a little more closely under high power, you can see that it is the walls that are parallel to the outside, which are thickened, and thereby provide support, similar to what you would find in the celery. Now, here is the cross-section of the stem of this plant up here in front, of the linum stem. It's not red on the inside the way I've colored it here. This is due to a dye. And what do I want to call your attention to are these white, almost translucent-type structures or bundles along the edge. These are the bundles of phloem fibers, the soft fibers. And if you look at them a little more closely, you can see the fibers. There's a very thick wall and a very little area where the cell is alive, if it is alive. And these are the bundles that are extracted or combed out of the linen plant and used by humans. Here's a sclerid. Sclerids, again, are more or less rounded cells. They have thick walls. And in plants such as water plants or camellias, where insects like to eat them, the sclerids develop sharp points on them. And many people believe that this is to retard insects. It gets stuck in their mandibles, and it prevents them from eating. Here's another sclerid from a water plant. And again, you can see these sharp points, which are believed to be deterrent to insect eating. And finally, I want to just show you the coat of, in this case, a peach pit, which also is very hard, like the walnut. This is of the ovary, the inner part of the ovary wall of the peach pit. And again, you can see that it is a solid matrix of sclerids. And this solid sclerids, let's see, clear. Well, it asked for my password. Can you put the screen up since I don't have one? Thank you. Okay, so these are the sclerids. Now what I want to do is to move on to what we're going to have as our first tissue type. That is our first group of cells in which the tissue consists of more than one cell type. And that cell or tissue type is, well, that doesn't want to clear for us, but we're okay. That cell or tissue type that we want to consider is involved with transport. And this tissue that we're going to consider consists of several cell types. Probably we can make these lights a little higher. Maybe. There we go. Okay. Several cell types. And for our discussion of the transport tissue, which, as I earlier introduced you to this term, we call this transport tissue the vascular tissue. And there are two sort of subcategories of tissue which make up the vascular tissue. And I'll write their names down, and then we'll talk about each of them individually. The first tissue type is known as the xylem, and it is involved with the transport of water. And the second tissue type which makes up the vascular tissue or the transport system is known as phloem. And it is involved with the transport of photosynthate, that is, sugars. Let's consider first the xylem, which is the tissue that is involved with the transport of water. And we'll begin by talking to you about the two cell types which are associated with the transport of water. And those two cell types are tracheids and vessels, or vessel elements. Now, for those of you who have studied animal or insect biology, you will realize or appreciate that these terms are similar to what are used in the breathing system or in the blood system of those organisms. People who studied the xylem in the 19th century also realized that the xylem was involved in the transport of water, and so they used the same terms for the cell types that were involved in the transport as were used in the insects or in the animals. Let's talk about these two cell types. First thing we want to say is that they are dead when functional. And we'll talk about what this means in a moment. Dead when functional. Now let's talk about the two particular cell types, and we'll begin by saying that tracheids are found in gymnosperms and angiosperms. Whereas vessels, or vessel elements, is more appropriate, talk about what that means in a moment. Found only in angiosperms. So when the gymnosperms evolved, they didn't have vessels. And when the angiosperms evolved, part of that evolution involved vessels. So let's look at each of these two cell types which make up the vessels and the tracheids. And let's first begin with the tracheids. These are elongated cells, narrow, closed end walls. And that's important. You'll see why in a moment. Closed end walls which overlap other tracheids. So let's just draw one of these on here. Here is a tracheid, a long, narrow cell, and we said that it overlaps another tracheid down below. Now, if the end walls are closed, and we said that these are involved with water movement, how does the water get from one tracheid to another if the end walls are closed? And what we can say without exploring it further is that there are holes or pits located at the end walls which function for the movement of water, through which water enters. How about a vessel element? A vessel element, we said, are only found in angiosperms, and compared to the tracheids, we could say vessel elements, which I'll just abbreviate as VE, are short, wide, and have opened end walls. So let's talk about how water moves through a vessel, and in a sense, how it also moves through a trachea. I'm going to draw for you one vessel element, which we'll just call VE. One vessel element. And it is stacked on top of another vessel element and on top of another vessel element. Now, with time, the end walls dissolve and the cells die and become empty. So we produce a structure like this with time, in which the end walls have dissolved, and we have this structure here. This structure, which consists of a series of vessel elements, is known as a vessel. And it is totally open. It is essentially a pipe which runs through the plant. And to give you an example of the openness of this pipe, I have a piece of stem from a plant in which the vessels are large. And I'm going to just put it here for those of you up front. I'm going to blow through this pipe at one end, and you actually can see air bubbles, if you look carefully, coming out at the other. Those air bubbles are coming out of air which I am forcing through the open vessel element through these dead cells. So this is the xylem, an important cell type. Now, let's consider the second cell type which makes up the transport system. That is the phloem. The characteristic cell type which makes up the phloem is known as the sieve element. And we'll talk to you about it in a second. And the important characterization of the sieve element is that it is alive when functional. That is, it has a membrane. Now, what I'd like you to do is I'm going to draw up here one sieve element. And let's pretend like this eraser is a sieve element. So it's going to go right over here. And what I would like to show you is where the word sieve comes from. Why do we call this a sieve element? What I'm going to do is, if I could take this one-dimensional or two-dimensional cell I've drawn on the board and bring it out of the board so that you're looking at the end of the wall. That is, your eye is down here, looking at the end of the wall. What you would discover on this end is a cell wall which has lots of pores in it. This is known as a sieve plate. A sieve plate, and this gives the name to this particular element. Now, let's talk to you a little bit about how the sieve element forms, because you can go ahead and put the screen down, please, because it has a very interesting development, and I want to make sure you understand it, especially because it's alive when functional. So let's talk about the formation of the sieve element. We start out with one cell, which we'll call the sieve element mother cell. And if we go through some time, what we find is that the sieve element mother cell now undergoes a asymmetric division. She has a nucleus, and we have an asymmetric, an unequal division to produce two cells, both of which have their own nucleus in them. This cell is known as the companion cell. And this cell is the precursor to the sieve element. And now if we may wait a little more time, what we find happening in this particular situation is that the nucleus of the daughter cell, or the companion cell, still persists, but that the nucleus degenerates. The nucleus degenerates in the sieve element precursor. Not only does it degenerate, but also much of the cytoplasm disappears. But the cell is still alive. It has to have some way of making repairs, of keeping its metabolism going. And the way it does it is that the DNA and the RNA, which is used to keep this cell alive, which now lacks a nucleus, essentially the companion cell controls the existence of its adjacent sister, which is the sieve element. So when the companion cell dies for some reason, if it is injured, the sieve element dies. Now, why do we have to degenerate the nucleus? Why do we have to make it disappear? Why do we have to have cytoplasm disappear? Again, this is to empty out, in part, the sieve element to allow for photosynthate to move through it. And we're going to talk a little bit more about that movement when we talk about transport in the plant. So here we have two different cell types which are part of the vascular system and which function in transport of both water and nutrients. Let's look now at a few of the different cell types. The first cell type is the tracheid or tracheary element, and this one shows you a nice comparison between tracheids, which are elongated cells with solid end walls. They're much longer than fibers, but the vessels and the vessel members or vessel elements are much shorter. Notice that they're wider and they have an open pore. And if we look at the formation of the vessel, we find that it occurs because the end walls begin to dissolve so that you have this open pipe-like structure, and all the cytoplasm has also degenerated, making this dead cell, which is functional in the transport of water. And if you look at where two vessel elements join each other, you can see this is an end wall which has been dissolved away, and through this end wall, water moves from one vessel element to another. Here are some pits between adjacent tracheary elements. Again, they're much smaller than the holes at the end of vessel elements, but through this, the water flows. Here is an electron micrograph of a sieve element or sieve tube element, and what I want to point out to you is that the cell is open. Here is the sieve plate in this particular region here, and next to it is the companion cell, the cell that is providing the information to keep the sieve element alive. Here is a sieve plate, and what I want you to notice on the sieve plate is the phloem is running in and out of the blackboard. These are pores through which the nutrients flow. and an electron micrograph shows you the sieve plate. One sieve element is here, another is there, and the nutrients would then flow between these pores. The sieve element, sometimes called a sieve tube member, is located here. Here is its more densely cytoplasmic companion cell. And then finally we have the series I drew on the board to you where the mother cell divides unequally to make the companion cell, and then the nucleus degenerates in the sieve element, and we have a relatively open sieve element. In this case, we have two companion cells, but at least one companion cell has to exist in order for there to be some communication movement through the phloem. Now, finally, we can leave the screen down, actually. That's good. Finally, what I want to do is talk to you a little bit about the last, but you can turn the lights up. Thanks a lot. What happens if you put chalk on your fingers, somehow it doesn't read my fingerprints anymore. So if any of you want to rob a place, put chalk dust all over your hand. All right, here we go. The last cell type I want to talk to you about is the epidermis. The epidermis is the outermost layer of the primary plant body. Plant body. And being the outermost layer, it is the first layer of cells which encounters all of the insults, all of the pollution, all of the changes in the environment that the plant sees. So the epidermis, in a sense, functions in protection. Now, how does it function in protection? Well, if you looked at an epidermal cell, what you would find, and let's pretend like this blackboard was a leaf, and you're looking down on the surface of the leaf, that the cells that make up the epidermis look like pieces of a puzzle that are sort of very irregular shape. Cells of the epidermis, pieces of a puzzle. They are very tightly joined. Very tightly joined. And they are so tightly joined that there's really no space between the individual epidermal cells as far as protection. Now, in addition to being tightly joined so that nothing can enter the plant or insult the plant from the outside, they also secrete, epidermal cells secrete a wax to seal more fully this wax forms the cuticle. And most of you have had experience with this wax. If you've ever had an apple and you try to make the apple very presentable and you shine up the apple, you're shining up the apple by essentially polishing it with the wax that the apple epidermis has secreted. This is the same material, and it's water repellent, the same material that is secreted by the leaves in the epidermis. Now, the problem a leaf has, which is so tightly sealed with a waxy layer over it, is that how is it going to get in carbon dioxide? Carbon dioxide is necessary for photosynthesis to occur. And so what the epidermis does is periodically on its surface, depending upon the species, it makes holes or pores. So let's look at a pore on the epidermis. And you're going to do this in the lab. Here we would have a pore on the epidermis, and through this pore, CO2 can go into the plant for photosynthesis. The problem, though, is that when CO2 goes into the plant for photosynthesis, at the same time, water can escape from the plant and potentially could dry out. So there is another type of epidermal cell which evolves, a new type of epidermal cell to control pore size. And that epidermal cell, here's our pore, Here's our pore. That epidermal cell actually consists of two cells, which we'll just mention now, and we'll talk more how they function later on. These are called guard cells. And they function to control pore size. This whole structure, guard cells and the pore, is sometimes known as the stomata or stomatal complex. And we'll talk more about how it functions toward the end of the plant section. Finally, I want to say that the epidermis not only produces the guard cells and the regular cells on the surface, but it produces a few other cell types. And the main cell type which I want to mention to you is known as a hair, or a trichome is the technical name. And these hairs or trichomes have many functions, but they usually are involved with protection. Hairs which are produced on the leaf frequently elevate the insect away from the surface of the leaf to prevent it from eating the leaf. Hairs also function in environments where light is very bright and where the photosynthetic mechanism could be damaged. They function to reflect the light away from the leaf. Here is an example of which hairs function in allowing the seed to be dispersed. This is cotton. Cotton consists of epidermal cells which grow out of the seed of the cotton plant and are then picked up by animals, for example, birds, who want to use the cotton for their nests or their lair, and in the process they distribute the seed. Finally, the last type of epidermal cell which I want to mention to you is that we've talked about modifications of epidermis on the shoot system, but there's also an important type of epidermal cell which comes from the root system, and that type of epidermal cell coming from the root system comes from the root epidermis. And that, here's the root epidermis, and what you will notice is that some of the epidermal cells actually have something growing out of them. This is a root hair. Comes from the epidermis of the root system, and it has a very important function in increasing the surface area for water and nutrient absorption. So these single-cell outgrowths from the epidermis of the root have an important function. Okay, we'll go to the last slides now. Thank you. Okay, here is a leaf, and what I want you to notice on this leaf is the epidermis, which are these small cells on the top, and then this kind of looks like a thicker layer. This is the cuticle, which is deposited on top of the leaf and prevents it from drying out. Here we're looking down on the epidermis. You can see the puzzle-like cells which make up the epidermis, and here is a pore through which the CO2 enters, and on either side of the pore are the two guard cells, which you can see a little higher power in this slide here, and in a scanning electron micrograph view in this slide here. We'll talk about how they work in a moment. Finally, I want to just point out to you some of the hairs. When you rub a plant and you get, like a geranium plant, you get a very nice smell on your fingers. That's usually because of an epidermal hair or outgrowth which has been broken and in which there is a volatile liquid, perhaps used to retard insects, but something which we humans like. Here is an epidermal cell with this secretion at the top. Here's another one with a secretion at the top. And again, this is probably used in retarding animals and insects from eating the plant. Here's the cotton we talked about. And if you look at the epidermis of the cotton seed before the hair grows out, you will see the individual epidermal cells. Each of these will come into a cotton fiber or cotton strand. The entire length of cotton is one single cell. And here's an example taken at about 20,000 feet, 15 to 20,000 feet in the Andes of a plant. Here's a guy over here. Nothing else is growing. This is a cactus, which is covered with epidermal hairs, and these epidermal hairs function to reflect the very high intensity of sunlight, which is found at this elevation, and also to prevent loss of water. And finally, the last slides I want to show you have to do with the root hairs, these structures which function to increase the surface area to allow a greater uptake of water. And here you can see an individual epidermal cell which has grown out to make this structure, which is known as a root hair, and which functions dramatically but has a very short lifetime. Okay, we'll see you on Wednesday then.